Good evening, everyone. Welcome to LumCon Science Talks. This week we have Dr. Jimmy Nelson from ULL. If you are a first-time attendee, just to let you know, you can interact with the three of us who are up on your screen at this moment in time by using your question function. If you are a return attendee, you know how that works. And everyone who's attended before knows I start with a stupid question at the beginning of these things. Tonight, no question. Tonight, I have a joke. So let's see if you can come up with the answer to this joke. And Jimmy, this one is especially for you. And Craig, I have one especially for you too. So we'll start with Jimmy. Jimmy, what kind of music should you listen to while fishing?
Oh, that has to be Jimmy Buffett, right?
Almost correct. It's something.
Almost.
Yes, it's actually something catchy.
Oh no, you didn't.
Bubble pop, I love that. Catchy tuna, thanks, Jonathan. Ooh, line dance. Line dance music from Jeff.
Oh no. Not.
You guys, bass music. Oh my gosh, you guys are funnier than my joke. All right, one more. This one for you, Craig.
Okay, here we go.
Ready? How does a squid go into battle?
I don't know.
Well armed.
Oh no. That is so bad.
Tori says soul music for the last joke.
I thought Jimmy was gonna say something like, you know, you gotta listen to like Jimmy Mullet or something like that.
That would have been better.
Awesome.
See,
Awesome.
you're quicker on the draw than. Yeah.
All right, you jokesters, it looks like everybody's got the question function down. And a few statistics for our audience. We have now reached 525 people through our science talk. So thank you so much, everybody, even if you're not attending tonight, for attending our talks. 9 of you will earn a challenge coin if you've attended tonight. 19 of you are within 3 talks of earning one. And we've had 150 people. Oh, there's the challenge coin. So those of you who have reached number 10 tonight, you'll get an email from me tomorrow asking for an address in which we can send you one of our research vessel Pelican challenge coins. So congratulations to those of you out there who've reached talk number 10. Those of you who haven't, keep attending and earn that challenge coin. With that, I will turn it over to Craig. Thanks, guys.
Thank you so much, Mert. Normally I start off this talk every week by reading off a script in which my statements are really prepared, but I thought tonight my comments should be more organic. When we started this at the beginning of the year, we didn't realize it was going to be so successful that we would have regular attendees coming back every week to join us. And I have to say that over this time, one of my favorite parts of every week, despite the world around us, this beautiful spot every week, every Thursday night with everybody attending and listening to some really amazing science, has proven to be a consistent highlight. And I want to thank all of our speakers over the course of this series, as well as all of you all attending and being so interactive with us. We really appreciate that. It's a real treat for us to be able to interact with you all in this way. Like Mert said, this is the 10th of our series. So, you know, and we have plans to keep the series going on in the future. Even after we sort of return to something that may resemble our lives before. And so we're really excited to continue this on, and hopefully in the future when we bring up our Blue Works, which is our new building as part of the new maritime campus in Homa, we'd like to start thinking about doing sort of public once a month sort of public talks in that space as well. And we hope you all will have the opportunity to join us there. As with a lot of excitement tonight that I get to introduce my friend and colleague, Dr. Jimmy Nelson. Dr. Nelson is a faculty member at the University of Louisiana at Lafayette. He's actually the Fritz Long BORSF Professor of Environmental Biology there in the Department of Biology. He's also a National Academy of Sciences, Medicine, Engineering, Golf Research Program Early Career Fellow, which is a huge honor. He got his PhD in 2011 from Florida State University in Tallahassee and was a previously research scientist at the NBL Ecosystem Center in Woods Hole. We were truly lucky to have Jimmy be one of the scientists in our state here in Louisiana. And Jimmy, thank you so much for joining us tonight, and I'll turn it over to you.
Hey, thanks, Craig. I really appreciate it. It's awesome to be invited. Thanks, Mert, for organizing everything, and for Craig for starting this up. I think it's a great thing. It's fun. I've tried to watch a couple of these. It's really awesome. And so if everybody can see the screen and things, I'm going to talk to you tonight about, can any just give me one indication that y'all can see the big cool fish on everybody's screens.
Responses are coming in. Yes, yes, yes. I see it. Yes, yes, yes.
Awesome. Awesome. Cool. Okay. So I am Jimmy Nelson. I am a assistant professor in the University of Louisiana Lafayette, and I study fish. And so tonight I want to tell you about a fish that I spent the earlier part of my career studying, which is just absolutely fascinating to me, and is the Gag Grouper that lives here in the Gulf of Mexico. And I have no doubts that you will find this animal to be as amazing as I do. And so here we go. And so right away, you know, I'm here to show you big fish. So we're not going to bury the lead. We're going to put some big fish pictures up right away. These are Gag Grouper. So on the left-hand side here, that is the biggest Gag Grouper you've ever seen. I know that because it is 3 pounds bigger than the world record, and that is me standing behind it. And on the right here is another picture of me holding a Gag Grouper. That's the largest female Gag Grouper you've ever seen. I know because we've caught lots of them, and that's by far the biggest one that we have ever caught. And these are some amazing fish. And I owe these fish actually quite a lot. I got to where I am today by studying them. And so I've had to, and so you can see some of the pictures of things that, some of the fish that we've caught and things that we've done. I'm gonna share some of the stuff I've learned about them. But the fact that we don't know a lot about this fish's life cycle, and we don't know a lot about what this fish does, even though it's very, very critical to a large part of the economy of the state of Florida and the Gulf of Mexico generally, allowed me to get to where I am in my career today. And so I've caught and killed more of these than I care to mention. I've actually done repeated surgeries on these fish and kept them for 2.5 years in a tank at the Florida State Marine Lab. And believe it or not, the fish that I kept for 2.5 years at the Florida State Marine Lab, we actually were able to donate them to the Ripley's Believe It or Not aquarium up in the Smokies. And so if you ever feel like seeing my fish that I worked on for a dissertation, you can head up to Gatlinburg, Tennessee, and you can see all 4 of them still swimming around in the aquarium there. I've done lots of chemical analyses on these fish and lots of analyses and all kinds of things. But along the way, I learned so much about this animal and just how incredible its life cycle is. And I think it's something that everybody should hear. And so I wanted to tell you a little bit about it tonight. So our story is here in the Gulf of Mexico. And so if people aren't super familiar, here's the state of Florida. And if you go west, we're going to go through the little brief coastlines they have here in Alabama and Mississippi. We're going to get over here into Louisiana. And so the area that we're talking about tonight is primarily here in what would be the northeastern part of the Gulf of Mexico, the area that extends into what is known as the Big Bend of Florida and along the shelf edge of the Gulf of Mexico in the sort of the northeastern section of the Gulf of Mexico. And I've lost my cursor. So in reality, so when these fish are born, when they're put out into the world, and so we're going to start at the beginning of these fish's life cycles, we actually know not a thing about the fish in the plankton, which is remarkable, as you will see, because this fish is not only a major fisheries fish, but also an important recreational fish throughout its range in the Gulf of Mexico. And for the longest time, we actually thought that this fish was in the plankton like everything else, and we tried to find some, but it wasn't until 2014, only 6 years ago, that we actually figured out that these fish are transported from their spawning grounds when they're larval fish in the plankton into their recruiting grounds by bottom currents. So they're born in 350 feet of water, and they immediately go down to the bottom where they are transported into the coasts along these bottom currents, and that we had not discovered until 2014. This fish has been being fished since the records have been being kept since the mid 1950s, and we did not know actually how these fish were being reproduced and being put into their nursery grounds until 2014. But here you are. Here it is. So this is a Mycteroperca. This is actually not the absolute picture of a gag grouper, but this is what it would look like. So this is a baby gag grouper. This is the smallest iteration that we have a picture of. And then this is what it would look like when it comes out of the plankton and gets ready to settle in its nursery grounds. It's this tiny little fish that's fairly translucent, and it's just barely beginning to resemble the form that it will when it's much bigger. And most often in Florida, these fishes land in seagrass beds. And in the Big Bend of Florida and throughout the northeastern coast of the Gulf of Mexico, it is covered in seagrass beds. And this is one of the major nursery grounds for these habitats, or nursery grounds for these fish. These habitats are very important for these fish. And like any place, when you move in, these new fish, they're welcomed warmly by the residents right after they arrive. They're greeted by the locals who are absolutely super nice to them and welcome them with open arms. They're prey for any number of predators from the very beginning. Anything that can put them in their mouth does, and they prey upon them incredibly. And that goes from when they're very, very small, and it's things like blue crabs and then as they get larger, it's other predatory fish, and then even when they get bigger, you see them in the guts of things like bull sharks and other predatory animals. But the reason that we find them in the seagrass beds is if they can manage to avoid all of those mouths that are after them, what you find is that these seagrass beds are so full of productivity of other prey items that the groupers can feed on that they grow really, really rapidly. And so when they first land, we find that they eat lots of little worms, and then they move on to shrimp, and then they move on as they get bigger and bigger to things like pinfishes and other little schooling fishes in the seagrass beds, and they continue to grow larger throughout the summer. And so they land in these seagrass beds in the springtime. They spend the summertime growing, getting larger, fatter, bigger, and then as the summer progresses, this is what they can look like now. So here are your gag grouper as I would call it the toddlers. If we're gonna humanize them, they're toddlers. This is what your toddler gag grouper is like. And then you can notice that, right, like from the pictures we saw at the beginning, they kind of look fully formed. They look kind of what they would look like kind of throughout the rest of their lifespan. And so, in fact, one of the names for these are greenies or grass groupers. Anglers catch them frequently in the summer times in the seagrass beds of Florida and in the western coast of Florida and even all around the East Coast and South Carolina and things. You can catch these groupers. And in fact, when back in the day, when the fisheries and things were first starting in the Gulf of Mexico in the late 1800s and things, these fish would actually end up in fish markets. They would get caught as juveniles and sold and eaten as grass groupers. And so before we knew anything about their life cycle at all. And so this is what they look like sort of at the peak of their seagrass residency time. And when they hit this stage in their lifespan, the seagrass begins to turn cold and the sort of temperate subtropical regions of the seagrass meadows, the Big Bend, get a little too chilly for our friends the groupers, and they vacate. And so they start to swim offshore after they've attained the size and start moving ever so slowly as they begin to mature out onto sort of deeper reefs. When I say deeper reefs, I mean sort of hard patch wrecks, reefs, hard bottoms that you find in the northern Gulf of Mexico throughout the early fall and into the late winter. You start seeing these fish show up on the reefs. And so after moving offshore as juveniles, they spend 3 to 4 years in these shallow water reefs, growing and then eventually becoming mature. After about 3 or 4 years from the time that they hit the seagrass beds, these fish can become mature at a length of about 60 centimeters, but for us Americans, that's 24 inches or 2 feet. Feeding primarily on fish. They're piscivorous. They like to eat fish. They ambush those fish. They hide in a little hole. They hide in a little crevice. They see a little fish swim by them, and then bam, they swim out and hit it and eat it. And they're sort of what we would call ambush predators in the field. And so, but one thing I forgot to tell y'all, which is actually super critical, all of the gag grouper that you've seen so far, with the exception of one of the pictures, is they're female. All of these fish are actually born female. They are what is known in scientific terms as a protogynous sequential hermaphrodite, meaning they are born female, and at a certain point in their life cycle, they will actually change sex and become male. And for gag grouper, that's actually a very complex process that we have only just in the last few years sort of begun to understand how that happens. And so, but for this fish, all of the ones that hit the seagrass beds and all the way through their life cycle for the first 5, 6, 7, 8 years of their lifespan, they're all females. They're all female fish. Even after they mature, they're going to spend at least another 6 to 7 years being female groupers in these shallow water reefs in these areas that we find in the near shore. After they mature, once they become mature females, after they're about age 6, 7 years old, in January, these mature females group together. They form these large schools, and they begin to swim further offshore. So they leave their home reefs in shallower water. They begin to swim together further offshore, where for the first time in their lives, many of them, they actually encounter a male. And so these males, the males, which we're going to talk a lot about in a minute, are known as copper bellies. But one of the most fascinating things about the gag groupers is that they aggregate together to spawn. So they get together in these big groups, and we actually have no pictures of gag grouper getting together to spawn. We know that they do it in behaviors from catch records and things like that. And so these are some examples here of, these are Nassau groupers spawning. And these are Goliath groupers spawning in big aggregations. So they all get together and then they spawn. Now, the interesting thing about gag grouper, but also a lot of other grouper species, is that what happens is that you have males and females. They actually do pair together to spawn. You have 2 fish that are supposed to spawn together. But most frequently, what you do see when we see video recordings of this is that other males. So here's an example of these are some Nassau groupers that are spawning. The actual male, the male in this picture is this male here that was the original initiator of the spawning event. And what you have here indicated by these arrows, the arrow here, are what we call sneaker males. And so everybody gets together, they spawn together, but as the dominant male is doing his thing with the females, and they start doing their dance and they go to spawn together, when the 2 fish get together and actually spawn, lots of sneaker males. And so here, this is a cloud. This is most of these fish in this picture of these Nassau groupers are actually sneaker males. The original run was done by 2 fish, and so you see all these other males come in and try to get, just a couple of bit of their DNA material to go on to the next generation. So even though they're not dominant, that doesn't mean they're not going to give it a good go. But with gag grouper, the fish that is most fascinating to me are what we call in the local language in the Gulf of Mexico are known as copper bellies. And so what you see, these are male gag groupers. All of these are male gag groupers. So you've seen this male gag grouper before. Like I said, it's the biggest one you've ever seen. And so what you'll notice is there's a distinct colorization on their stomachs, on their bellies. And so they get this. And if you're up close and looking at it, it does. It very much resembles a tarnished copper penny. It has this sort of dark blue-green aspect to it that looks like a tarnished penny. And so these guys, what they do, when they're males, so how do gag grouper decide or begin to determine when it's time to change sex? It is not a size selectivity thing. It isn't like if you're all of a sudden you go from 2 feet to 3 feet to 5 feet, then at 5 feet you're a male. It isn't all of a sudden when you reach a certain, if you're the most the heaviest fish, for example, or the oldest fish. It isn't about that. These fish actually have a social structure that determines when a particular individual will begin to change sex and become male. And these males are absolutely fascinating because what they do is they hold and defend a territory. And this will be very important to our story later, because these males are sitting on a place, they're defending a place, and which is theirs. And so when the females come, when they aggregate and come out to spawn, these females come to that place, and then the males, we call them harems. I hope, if you don't know what a harem is, Google it, feel free. But it's they gather females into large groups together, and then the biggest and the baddest of the males have the most fish in their harems, and they spawn with the most of the females, and they monopolize the spawning opportunities. And so just for a minute, what I want kind of everybody to think about is to get to the stage of these copper bellies, and to get to the stage of being an adult male gag grouper. You went from being this little guy here, eking out an existence in the seagrass beds, and then you dodged all of the things and the pitfalls and the traps and the mouths and the hooks to make it that far. And so one of the things I think about all the time is that when these fish are spawned, when I go and do my fishing, I'm a little different. I'm a little weird to understand. I'm a marine biologist. When I go fishing, I think about different things than maybe some other people think about. But what I think about when we catch these fish offshore, right? These fish were in the plankton. They were larvae for 2 months, and they survived this hundreds of kilometers or 100 miles across the Gulf of Mexico as plankton. And then they land in the seagrass beds where everything wants to eat them, and they survive where everything wants is trying to eat them for 6 months during the summer, right? And they grow and they eat and they get bigger and bigger and bigger. And then and then finally they're about a foot long. And then after they're foot long, the place that they've known for their entire lives and the place that's fed them becomes sort of inhospitable by being cold. And then they have to sort of swim offshore, and they have to find a new place to be where they have to find new food, and they find the new food and they eat the new food. And for 3 or 4 or 5 years, they grow in these reefs until they're mature. And then finally, once they're mature, they get into these spawning aggregations, and for their instincts and things, they don't even really understand why they get together, but then they get together and they swim offshore for the first time, where they actually encounter males and their instincts take over, and they in these spawning aggregations, and they spawn to close the cycle. And so when you're talking about a fish, and so I'll tell you, so this fish here, I aged this fish. At the time, I was 24 when this fish was caught, and that fish was 28 years old. That fish was older than I was when I caught him. And it was just sort of, when I sitting there looking in the microscope, aging this fish off of its otoliths, it sort of like, crystallized for me, like all of the things and all of the, I had not even been alive as long as that fish, and all of the things that it had to go through when, if I think about it for a minute, I don't even really want to go, drive to, the airport to get on an airplane. And I don't think I could survive like a car ride across, Louisiana with my mother-in-law, let alone, all of the things that these fishes had to go through. And so it just amazes me, all of the things that they actually get through, and then we actually have this incredible fish that supports this incredible fishery for all of us for us to enjoy. And even though we don't often know it when we're putting it in, there's all of this background and all of this story to these animals that we don't actually consider. But these are a fishery fish, right? So this is at the end of the day, you know, we go out, we catch fish, and we catch a lot of them. And so this is the Gag Grouper fishery in the Gulf of Mexico since 1963. The newest stock assessment I could find was up to 2014, but I can tell you the numbers are fairly similar. And so just sort of what I want you to get out of that is that we had some pretty good times here, as most did through the 1960s and 70s. And then in the 80s and in the 90s, the fishery was doing pretty well. And then as of late, the fishery has done not so well. And so one of the issues that occurred during his time was that from the 1970s to the 1920s, the year 2000s, one of the things that was noticed was that as we were fishing at this sort of higher level, the number of males in the populations went from 20% to 2%. And so this right here is sort of an interesting question, right? And so if why would the natural state be if you're this sequential hermaphrodite, meaning all of your individuals are born female, and then after their some time and some pecking order things, they become male. The reason that we have this only 20% of the fish being male is that if they have harems, they can spawn with many fish, right? So you want those dominant fish, the ones that ran that gauntlet, the ones that are able to make it all the way through. Those are your biggest, baddest fish. They have the best genes that you want to pass on. And so the sperm that they produce is actually not very energetically, or it doesn't take a lot of energy to make the sperm, but it takes a lot of energy to make the eggs. And so the strategy here through evolution and adaptation would be to have fewer males, but that fewer males could help reproduce the population, because many females can be fertilized by a single male, because they reproduce in this sort of haremic way with one or a couple of males being able to reproduce with many hundreds of females. But on through the 70s, as we continued to fish this fish, what we noticed was that sex ratio went down from about 20% down to 2%. And so now, not now, but at the time when this was being noticed, that makes you nervous, right? And so now you worry that even though you have females that are there to spawn, if they come out to places where the males have typically been, now you worry that there won't be enough males there to actually take care of enough females, and then to continue to produce the eggs and produce the larvae that are going to keep your fishery sustainable and keep producing the fish you need to keep the fishery going. And so in the early 2000s, the solution for late 1990s and early 2000s, the solution the National Marine Fisheries Service came up with, as part of NOAA, was to create areas of no fishing, or marine protected areas, MPAs. What these are areas of this is in the Gulf of Mexico, so this is the port. This is Panama City. I put this map up here because that is where I grew up. I grew up in Panama City, Florida. So this is the Panhandle of Florida. Most of you know the beaches of Destin and things, which are just right over here. And so just about 100 miles south of where I grew up, there is a marine protected area now. And it's a 10 mile by 10 mile square that protected Gag Grouper habitat. They also established other habitats to protect different types of groupers. These edges and steamboat lumps habitats were designated to protect Gag Grouper, but also other species of grouper in the Gulf of Mexico. And so what these MPAs, or these what they were designed to do, was to create areas of no fishing. So you had no fishing pressure, and so you had you had a place where we could do research, and we could begin to try to understand this, these really complex life cycles that these fishes have, so that would help us better manage these fisheries and to make them sustainable into the future, right? And so the reason that this area was chosen, particularly for Gag Grouper, was it would contain these habitats that for some reason, I mean, for lots of reasons, but these habitats here along this, what is essentially the old, what would be edge reefs that were, when the sea level was lower a few thousand years ago, these edge reefs, these were actual growing reefs. They were growing like reefs that you would see in the Keys, in the Florida Keys, and they were adding height. And so Gag Grouper liked this relief. And so when the ice melted and the sea level rose, it drowned these reefs. And so they're no longer living reefs, but they still have this very high relief. These are drowned patch reefs from pre-ice age. And the Gag Grouper adapted these areas and used these areas as their spawning habitats. So this edge of the continental shelf here is actually a very critical habitat for where the male Gag Groupers go and create their habitats. And so, or create they use this habitat for spawning. And these male Gag Groupers stay on these sites, and they designated this area as an area where there'd be no fishing, so that we could then study the fish in the habitat without fishing, to try to understand what it was, the mechanisms behind this change in sex ratio. What is it actually behind how Gag Grouper actually changed sex? What are some, what are the dynamics between, when reproducing females, do reproducing females stay? Do they go? Do they come? Where do they come from? And so that was sort of the charge that we had when I was in the lab and throughout some of the work I did to get my PhD. Now, if you're going to study a marine protected area, one of the last things you want to do is catch and kill all the fish in your marine protected area, right? If you're trying to understand dynamics without fishing, you as the scientist cannot be a fisher. You have to come up with novel and a novel. I mean, at the time they're novel because they're, trust me, as a person who does fisheries type features, the easiest thing to do is to get on board of a fisheries boat and to cut the fish, kill the fish, take what you need from it, and do your analysis, right? But if you're going to work in a marine protected area, you will never be able to actually understand the difference between the effects you have as a fisher unless you do non-consumptive sampling. So we had to be very creative in what we did. We had to be able to get the fish out of the water and then bring it up, take the things that we needed to take to learn about its age, about its life cycle, about its life history and things, and then be able to put it back in the water and it would survive. And so that's kind of a problem, and it's a problem because of an anatomical structure that groupers have. This is a picture of a red grouper, and if anyone's been deep sea fishing before and caught groupers, this is a picture of a red grouper, but I could find a picture. I don't have one, but I could find a picture eventually of a Gag Grouper, the same thing. And you see this little bit here it looks like its tongue sticking out. That's actually its stomach. And the reason that happens is that these groupers and lots of fishes have a structure in their body known as a swim bladder. And if I have any divers in the audience, a diver that uses a buoyancy compensator or a jacket that you fill with air to help you sort of stay neutrally buoyant in the water so you don't really sink and you don't float, but it helps you keep a nice even keel in the water, that's what these swim bladders are for the groupers, right? They are the buoyancy compensators for the groupers. They allow them to stay in the water, to stay upright, to maneuver, to hang out in the places that they need to be and conserve their energy without having to swim a whole bunch, right? But if you're a diver, and I can explain boils a little bit, but right, so if you're deep in the water, these gas bladders, they can't exchange quickly. They're not like our lungs. They have a very thin membrane which is what they exchange gas with, so they can't exchange quickly. So if you hook a fish on a line and you quickly bring them to the surface, that gas bladder expands. As that gas bladder expands, it pulls on the organs of the fish, and most unfortunately for our friends, the groupers, the lower part of that gas bladder is connected to their stomach, and the upper part of that gas bladder is connected to their liver. So when you bring that fish up, if that gas bladder expands and ruptures, and it sends the stomach through the mouth of that fish, it actually tears their liver in half. And so even if you bring them up, and even if you put them back, often what would happen with these gas bladders is that they would rip their livers in half, and then those fish would bleed to death and die. Even if they swam back down, we did experiments that showed that if you had this barotrauma, it's not the bends, it's different. It's like if you held your breath, if you took a head off a regulator, held your breath, and then came up. If you had that barotrauma, it was very, very unfortunate for the fish. So we had to come up with a way of catching the fish at depth, bringing them up, relieving that gas pressure, and then bringing them up to take all of our samples. And so one of the coolest things I've ever got to do in my life was to pop fish at 150 feet in the Gulf of Mexico. And there's no other way to describe it than to show a video. And so here you go. This is me, and I hope you can see it, popping fish in the Gulf of Mexico. The first thing you'll see is a shot on that's a snapper there against it. And here's a grouper, and we hit the grouper, and you see the bubbles of the fishes. As the video plays, you'll see bubbles coming from the fishes. And so what we're doing is we're using a little pole sphere, a Hawaiian sling in the parlance. And so as you hit these fish with the gas bladders hit the fish in the gas bladder in their swim bladder, that relieves that pressure. And then what we found through experimentation is if we did this, we were able to bring the fish up from the bottom. We were able to take our samples, and we were able to put the fish back alive, and they would stay alive and to be caught again or to live out their lives normally. And so what that allowed us to do then was to take the samples that we needed to do to sort of figure out some of these these mechanisms that we had questions about how the fishes changed sex, how old the fishes were, how they did what they did, where they were going, and how long they were staying. And so one of the things that we did was that we were able to do these are slides of gonads of gag groupers. And so by repeatedly catching fish at particular times of year and putting them back down, we would take histology slides. So all histology is a very thin section of a tissue. And so when we would bring the fish up after they're caught in the trap and then vented, we would take a very teeny piece of their gonad, of their reproductive organs, and we could then section it very thin, and what you're looking at are cells. And so these are the cells as they typically are in a gag grouper female, which remember, they're all born female when they're not spawning. As they go to spawn, these cells get larger, and they get what is called phylogenetic. They get yolk in the cells, and then eventually these cells, what you see on this bottom side, these empty areas are where eggs used to be, and these eggs have been lost. So this is a fish that has spawned, right? So this is the typical cycles you would see for a female. They would have these sort of primary growth eggs. The eggs would get a little bigger, and then as the eggs got really big, they would spawn them, and you have these pockets of empty egg space. What you're seeing here is this is a female that is beginning to undergo the transition from being female to being male. And so what literally happens is these egg cells begin to break down and actually begin to literally transform into sperm cells. And then eventually what you see is that these sperm cells become more and more conglomerated, and then eventually these sperm cells they start breaking down, and you get these big ribbons of sperm, and then the fishes officially transition to male. And one of the most important things that we learned is that this happens over a year for the fish. So if the fish begins the transition, it takes a whole year before that fish can come around again and reproduce as a male. And so this is sort of a critical piece of information that I'll explain later here about that was important. The other thing that we were able to do then is age the fish. And so by taking spines and raise the fish, just like you would have in the ring of a tree, the rays, so the spines are the pokey parts, the hard protective parts of the top sides of the fishes, that if you ever picked up a catfish the wrong way, you know what I'm talking about. It's the things that would stick you in your hand. If you take those structures and you make a really thin section of them, you can actually see rings in them like you would a tree. And so here's the core of the ray. Here's 1 year, here's 2 years, here's 3 years, and then they're moving on to year 4 here. And so the combination of the analysis of their gonads and the analysis of the ages, we learned that it took about 10 to 12 years for these fish to become big enough and mean enough to start transitioning into males. And once they started transitioning into males, it took them a whole year to come around to actually be able to spawn once they had become males. So how does this help us understand what happened and why the sex ratio went from 20% males down to 2% males? Well, remember, the males hold spawning territory. So when when the females come out, there'll be a couple of males here, right? And then the females come out, and then they spawn together, right? They'll all spawn together, and then the females can go back home. Well, if one of the males dies, when the females come out, one of those females will begin to transition over to becoming a male. And so the female will join the males in the population. She'll leave the female population and remain and stay, right? And so then that's fine. So then the females can all go home. But the way we fish this fishery is that fishermen come back to a spot. They come back to a place. We put that number in the GPS, and the fishermen will then come back to that place and fish it, right? And so they'll come back, and when the females, there's nothing but males there, what the fishermen will do is preferentially, they'll take the males away. The males will now be removed, because the only thing on that fishing spot when the fishermen go out there are the males, because the males stay on their territories throughout the year, even though the females go back to the places from which they came. Now the problem is that the next year when the females come out, now there are no males, right? There are no males for them. So the social structure kicks in, the baddest females stay, right? And so then they stay, those females change sex, but they can't, right, in the same year then go and reproduce and then make more baby fish. So all of the females that were there, they have to go home, and the cycle repeats itself. And so what we hypothesized was that this cycle of males being put back in the population, being caught by fishermen, and the females coming out, missing spawning opportunities, and then trying to transition. But in the time that it takes them to transition, they're then caught by fishermen. That's one of the mechanisms that's our hypothesis for how that sex ratio went from 20% in the 1970s down to 2% where it is today. And so that is these marine protected areas, right? These marine protected areas are places now where these fish are not going to be fished, and they get to go through that life cycle and go through that sex change and complete it without the pressure of fishing, removing those fish. And we've actually seen some evidence that as these areas remain closed to fishing, that they do increase their numbers. But this is the one thing that I sort of want to leave you with, right? So these are fisheries fish, right? And so we put these marine protected areas in to protect this fish as we see it and as the fishery is. And we want to keep things the way they are, keep them or make them better, right? And so this is anybody who's ever gone on a charter fishing trip. Here's a charter fishing trip that was done in 2020, literally a couple of weeks ago. This is a pretty good fishing trip in the Gulf of Mexico, most people would say. Here's a few gag grouper on the board, right? Here's a few red grouper. These are red snappers. There's a scamp right here, and these are red snappers on the ground. That's a pretty good fishing trip, right? Like, I think many of you in the audience would probably agree. You wouldn't mind being on that boat that day, right? This is a fishing trip in 1968 from the same dock, from the same area. This is a Warsaw Grouper. It weighs 250 pounds, or sorry, 150 pounds. These Warsaw Groupers are large fish. There's a couple of gag groupers on the deck. Actually, there's one right there, which would be bigger than any of the gag grouper on the previous board, right? And so this trip occurred, the previous trip that you saw, this trip happened over 12 hours. The trip here where these fish, it took the entire day, sun up to sundown. This trip occurred over 4 hours. This is 1/3 of the time in 1968. And so one of the things that I want to leave you with is that as we do these management things and as we try to change our fisheries, we run into this problem of what's known as shifting baselines. This is the same pegboard in South Florida in the 1960s, the 1980s, and the 1990s. The folks here in the 1990s, if they had a trip like this, would never have put this stuff on the board in the 1960s, right? And so as the old generation grows old and they pass away, our perception of what is good, right, changes and shifts. And so if we want to manage our fisheries and if we want to make our fisheries better, we really need to understand what an ecosystem is actually capable of producing, right? What we can get out of that system, like what are we restoring? What are we trying to get back to? What can these systems really be versus where they are right now? And is going back, you know, good enough? And so what I just want to leave everybody with is, like, you know, we're doing okay. We're managing our fisheries. We're actually not doing a terrible job managing what we have, right? But is what we have what we want? And so the questions, the sort of the question that I have is that we manage our fisheries well, but could we manage them better? And could we, you know, get back to things as they actually were? You know, if this was our goal, if this was in our memory, would this still be okay? So I just want to thank everybody for their time, and I really appreciate letting me talk about my favorite fish. And I'm happy if I have some time to answer any questions y'all have. And thanks, Craig and Mert, for letting me do this.
Great, Jimmy. Thank you so much for a great talk. And we'll just let people process your last few statements as they're typing in their questions. If I can get you to stop sharing your screen so we have a better view of you while you're answering questions. We have a lot of questions coming in.
Did that work?
So we'll start with some of the earlier ones that came in at the beginning of your talk. So We Hunt has two questions. He would like to know how old the larval grouper was that you showed, and he would like to know if grass groupers are kept by fishermen or if they're let go.
That little fish was about 60 days old. So it takes them 2 months in the plankton. I don't even have a picture of what they look like in the plankton because we've never actually caught one. But that's what they look like when they first come out. So that's 60 days. Grass groupers used to be kept. They used to be kept and eaten and grilled on the beach. But no, it is absolutely not allowed now because those are juvenile gag groupers that need to go out. Yeah, back in the day though, it was totally fine.
Nowadays, they have more important things to do.
Right, they need to go on.
Becky was extremely interested in the life history and reproductive behaviors of the groupers, and her biggest question was, do all groupers do the same life cycle as the gag grouper, or is that just species specific?
It is no, they do not. So there are anything you can possibly imagine with your brain, fish do it in terms of reproductive strategies. Most groupers have some form of sequential hermaphrodism. Some are born 50-50. We think that Goliath groupers are born, the really big ones that you see in South Florida, we think those are actually born 50-50. But again, we think, like I said, we even though we fish these fish, we actually don't know the whole story. Some fish are born the other way. Some fish, like snooks and things, are born first male and then switch to female. So no, in groupers, it's the entire suite. Some are female first, 50-50, or male first.
Along the same lines, Tori would like to know if it's possible that once a change has been made, can they change back to the original sex?
That's a great question, and they do not. So that was one of our questions when we were studying them. They do not change back. They are incapable. What happens is they completely change their hormones and literally rearrange their reproductive organ, their gonad is completely changed.
Okay. Becky has lots of great questions. Her next question was whether the seagrass beds that you were talking about were impacted at all by the 2010 BP Deepwater Horizon oil spill.
The specific seagrass beds in far eastern Gulf of Mexico were not impacted. There were seagrass areas and submerged aquatic vegetation areas in Louisiana and Alabama or in Mississippi that were impacted. Now, and that's a great question, Becky, because those seagrass areas are absolutely critical for many species. We must have areas like that, or else we will not have groupers like that we have. That's a really good question. development on the coast, building houses, building condos, poor water quality, oil spills, all of those things are not good things for seagrass beds, which are super critical to fish.
Great. Joel's question has a lot to do with your methods of getting air out of the swim bladder, and he would like to know how do you know they survived that, and if there was any internal damage?
That's a great question. So we did months of experimentation about how to do this. One of our experiments was this is how little we knew about them. We actually thought it was the bends. We actually thought that if you just kept them in a cage for a while and brought them up slowly, that you could bring them up slow and to be fine. It doesn't work that way. It takes them months to get rid of the gas that way. There's a whole physiological thing, and I'd love, if you have questions, you can email me and I can explain the whole thing. What we learned was fish, and if you think about it for a minute, it's actually not that crazy. Fish are actually really good at healing puncture wounds. Because if you get bit by a shark, right, you got to be able to heal that up. And so we poke a tiny hole in them, and by letting that gas out, we would put those fish back down and we would leave them in the cages, and we experimented. We would poke them, leave them down for a day, 48 hours, 1 week. We sat out offshore for 1 whole week with the fish in the cage and brought them back up, repopped them because we had to, because the fish had actually healed their swim bladders within the week. And then they were alive in the cages. So yeah, they are able to survive it, recover from it, and live. And we ended up tracking a fish that we had popped for over a decade, and the battery ran out on his tag. So yeah, they do live. Hey, you have to have good pokers now. If you have bad aim and you hit them in the wrong place, it's no good. But if just the swim bladder and a little hole, they actually do really, really well.
Great, thank you for that. Jonathan's question is about regulations. So what is the current, so I'll read his whole question. Sorry.
No, you're good.
I can't even read a question. So a single very large female grouper be more valuable reproductively than many small female groupers. What current legal regulations do you run into?
That's a super on point question. And it's a very sticky question, which is one of the reasons that we use MPAs, right? Or the protected areas. Because if you bring a big, you don't know when you catch that grouper, right? If it's legal, if it's big, if it's little, you just bring them up, right? And if you bring them up and the swim bladder ruptures and they have bad trauma, they die. So you can't do size limits. You can't do bag limits. That doesn't really, once the fish is on the hook and brought up, the damage is done. And so that was that's one of the arguments for having marine protected areas, because you're big, we call them BOFs in the field, we call them big old fat females, BOFs. And your big old fat females are the ones that produce many, many, many millions of eggs more than your little females. And so those protected areas, one of our hopes was that our BOFs were going to stay in that protected area, right? Because as they get bigger, they move further offshore. And so what we hoped is that it would protect the males, but would also protect your big fat females, who, yes, absolutely, he's absolutely right. Your big fat females produce millions of more eggs than your little females. Sorry, little females, don't get upset.
Jonathan also has a question about whether an edge reef is the same thing as a drowned reef.
A drowned reef is a reef that was being actively built, so I would call an edge reef a reef that is currently still active, being built. These drowned reefs were active during 10, 15, 20,000 years ago when the water was shallower and they were in the sunlight, but now they are no longer in the sunlight. They're dead. And they just, but they did grow. And so the fish like to hang out on that relief that was the vertical habitat created by the reef growth that's no longer happening. So they like to be in deeper water, but they like to be on stuff that's high, changing. Yeah. That probably
Great,
is not a succinct answer, but
I think you got it.
it's science. It's complicated sometimes.
He says thank you. Joel's question is, are there bag limits on the number of groupers that can be caught per day?
Yes, it is 5 in the state of Florida right now. I don't know what it is in Louisiana, but it 5. The answer is 5 per day per person per day.
Do you think that's too high?
I mean,
Sticky question. I retract
Right.
the question.
No, I mean, I'm not a fisheries biologist, but that's a lot of grouper for people. If you ever go and look at any, I mean, go down to Venice, go down to Cocodrie, you know, like y'all know, there's lots of people that go out and fish every day, and people, you know, people like to put a lot of weight on, you know, these big commercial fishing boats, but like there's a lot of people doing a lot of fishing, right? It's a lot of hooks. It's a lot. We like to fish as a species.
Joel also has a sticky question. I'm sorry, I'm throwing them all at you tonight.
I love them. Let's go. Let's do it.
Is sustainable fishing really possible?
That is a great question. Okay, so it takes a cow about 18 months to become marketable, to produce 1,000 pounds of meat. It takes a chicken about 6 weeks to produce a chicken, right? Like from the egg. And it takes a gag grouper about 8 years to produce 6 to 8 pounds of meat. So the answer to your question is there is absolutely a sustainable level of fishing that can occur on those fish. Are we at it? I don't know. But it is not going to be something that supports a cod or a pollock or a fish sticks at McDonald's type fishery, right? Can you support a regional, recreational, small commercial for like local grouper sandwiches and things? I think yes. Can you export big to Chicago and New York? I don't think so. That's a politician's answer, Joel.
Arturo has a question about what methods were used to track newly spawned groupers to their nursery grounds.
Here's the methods that were used. We dropped neutrally buoyant, they're ping pong balls, but they look like ping pong balls, but like they were filled with different oil to make them either buoyant or not buoyant. And we literally dropped them all. They're biodegradable. They're not plastic. They were all, it dropped at different levels. And we actually had to look for the current that brought them into shore. And that was how they were found. Well, the current was found because we thought for the longest time it was on the surface. But in fact, it's actually on the surface, the wind blows the water the opposite direction, which means that the water on the bottom is flowing in to fill in the gap. And that is how the groupers get to shore during the spring.
So those were passive trackers.
Passive trackers, lots and lots of passive trackers, and modeling, computer modeling.
Awesome.
It was super awesome, super awesome. I went to grad school with the guy that published the paper. He's a super nice guy too.
Michelle has a question about what your thoughts on the future of deep water fishing are, given that what you said about barotrauma. Sorry if I pronounced that wrong. We have descending device requirements now, but it sounds like that's not solving the problem.
It doesn't solve the problem. It doesn't. And I mean, the solution to deep water fishing, in my opinion, as a professional scientist, is areas that you cannot fish. If you have areas that you cannot fish, then you can have areas like I don't understand the disconnect. We've done this on land for hundreds of years where we have preserves. Preserves are not like a crazy concept in our social structure. I don't understand why there's such a crazy concept in fisheries. And the ocean's way bigger than the land. There's 3 times as much ocean compared to the land. So you're giving up like not that much. And I think that they could, and I'm not going to get on a rant, but I just feel like The future of deepwater fishing is marine reserves and places where we just don't fish.
Very good. Back to We Hunt. We Hunt would like to know how difficult it would be to collect fertilized eggs.
Very, very difficult. We have tried and tried and tried. We literally just tried to take pictures of them in a spawning aggregation, and we have yet to do it. Hey, if he can figure out a way, he'll be famous, because like we've yet to find it.
You just challenged We Hunt, which is not always safe.
I would love it. Let's go. Let's do it.
And we're going back to Becky, who is an excellent question. Is anyone culturing Gag Grouper or any other grouper? Maybe even for research?
They aren't. You know why? Because they're a terrible culture fish. They take forever to become mature. Like they're sequential hermaphrodites. So you have to keep one of them if you want to like keep your cultivar going. You got to keep them for a decade. No, they are not.
And Jonathan says he had a chance to work with Grouper at Woods Hole, and he thinks that their social hierarchy is very interesting. One of the things that he found very interesting while he was working with them is their learning capacity. Did you encounter situations where you were impressed by their learning capacity?
So absolutely. So I kept, and I wouldn't actually normally tell the story, but to a broad audience, but why not put it on the internet for all to see? I kept 4 of them in a big tank, and I did experiments on them. They are some of the most socially structured. They had places they like to be. They had routines in the day. One of their favorite thing to do when I would feed them, one of their favorite things to do was to herd fish into I would feed them live fish. They would herd fish and then do a circle around the tank so that they could do a darting motion to eat the live fish. They would herd it into an area where they knew it would stay, and then they would come around so they could like ambush it. And just for the thrill of it, or I have no idea why they would do that. They have complex social structure. They had there was a boss fish. We had a male, and the male kept he would literally place he would move the females around the tank. I think this is one of the most interesting things that's very understudied just generally in science. Is like fish have very complex social structures, and it's a fascinating thing to me. My personal science doesn't do it, but it's observing them for 2.5 years. They had personalities, they have social structure, they are 100% aware of their surroundings and their things. They have habits and routines. Yes, they're super complex animals. I mean, that's kind of why I wanted to give the talk the way I gave it, because it's just a super complex fish.
Jonathan says he totally agrees. Yep.
Cool. No, I mean, they're incredible animals. Incredible.
Yeah. We noticed some really amazing things about the grouper we had at LumCon for a very long time.
They can live for 50 years, right? Like they pick up some stuff. Like they're not yeah.
Yeah, we had ours for 10 or 12 years, and we were definitely being trained.
Yeah. Absolutely.
And that is all the time we have for tonight. I hope you guys really enjoyed this talk. Jimmy, it was wonderful. Thank you for talking about such an amazing animal that we have in the Gulf of Mexico. I can't begin to thank you enough. That was a great talk. And for all of you who have attended, thank you again for coming back. And for those of you who are new, see us next week. So we'll be back next Thursday with our talk. We'll try to get the registration information for that talk up on our website as soon as possible. Like always, Jimmy's talk has been recorded and will be posted to the same page on our website. So if you want to rewatch or have somebody who wasn't able to attend tonight we'll have that recording available. With that, I will say, have a great night, have a fantastic weekend, and we'll see you next week.
Thank you.
Thanks, Jimmy.